Welcome, scholars of science. Today we are diving into the fascinating world of enzymes, the cell's microscopic multitaskers. Without enzymes, your breakfast cereal would still be undigested by lunchtime. These biological catalysts make life possible by speeding up reactions that would otherwise take centuries. Let's explore how they work, how they are structured, and why they sometimes fall apart under pressure, literally. Enzymes are proteins that act as catalysts, helping reactions happen faster without being used up. Think of them as the biochemical speed boosters. Each enzyme has a unique shape that only fits specific molecules, much like a lock and key. Without enzymes, most reactions in your body would crawl along too slowly to sustain life. They're the ultimate behind the scenes workers, keeping your cells running smoothly. The active site is the enzyme's workbench, a specialized pocket where substrates bind and reactions occur. When a substrate enters, it forms a temporary enzyme substrate complex. You can imagine it like a handshake, brief but productive. The enzyme lowers the activation energy needed for the reaction, making everything more efficient. Once the reaction is done, the product leaves and the enzyme is ready for its next customer. All chemical reactions require some energy to get started, known as activation energy. Enzymes act like helpful teachers, giving the molecules a boost to get over the barrier. Without enzymes, this barrier is like a wall too high to climb. With enzymes, it's more like a gentle ramp, smooth sailing. This is why your body doesn't need to heat up to 300 degrees Celsius just to digest a sandwich. The lock and key model was one of the first ways scientists explained enzyme action. In this model, the enzyme is the lock and the substrate is the key that fits perfectly. It's a great visual, though a little outdated, kind of like your old flip phone. Still, it shows how enzymes are specific. Only the right substrate can activate the enzyme's function. One wrong fit, and the reaction simply won't start. Modern biology prefers the induced fit model. Here, the enzyme actually changes shape slightly to fit the substrate. It's more like a hug than a handshake. This flexibility allows enzymes to be more dynamic and efficient. It also explains why enzymes can sometimes handle slightly different substrates. Flexibility really is key in biochemistry and in life. Enzymes are sensitive. They like things just right. Temperature, pH, and substrate concentration can all affect their performance. Too hot and they unravel. Too cold and they slow down. If the environment strays too far from their comfort zone, the enzyme denatures and stops working. Even enzymes need good working conditions. Temperature and pH both have Goldilocks zones for enzymes. At the right temperature, they work efficiently. Too hot, they melt like butter in the sun. Too cold, they're as sluggish as a sleepy sloth. Similarly, extreme pH can disrupt the enzyme structure, so each enzyme has its own comfort range, just like people with their favorite climates. Sometimes, molecules called inhibitors sneak in and stop enzymes from working. Competitive inhibitors block the active site. It's like someone parking in your driveway. Non-competitive inhibitors attach elsewhere, changing the enzyme's shape and shutting down activity. Either way, they prevent the enzyme from doing its job. Some inhibitors are used as medicines, while others are poisons. Chemistry's version of handle with care. Enzymes often need a little backup. Cofactors, like magnesium or zinc, and coenzymes, like vitamins, help enzymes do their jobs. Without them, even the best enzyme can't function properly. Think of cofactors as the tools an enzyme brings to work. No wrench, no repair. So, yes, when your teacher says, eat your vegetables, your enzymes silently cheer. Enzymes aren't just busy inside our cells, they're all around us. They're in laundry detergents breaking down stains, in food processing turning milk into cheese, and in medicine helping diagnose diseases. Humans have learned to harness these tiny powerhouses for countless uses. From brewing to biofuels, enzymes do it all. Next time you clean a shirt or eat yogurt, thank an enzyme for its service. Let's wrap up. Enzymes speed up reactions by lowering activation energy, and their structure determines their function. Their activity depends on pH, temperature, and inhibitors. For exams, remember to link enzyme structure to its specific function and reaction type. Enzymes truly are the biological VIPs, very important proteins. Keep studying, stay curious, and let your brain's reactions stay exothermic in a good way. That's it for now. Don't forget to subscribe, like, and check out Dr. B's tutorials on Amazon for full AP study guides and IB study guides. See the first link in the comments below. For more tutorials and test prep videos, subscribe to our YouTube channel, Dr. B's Tutorials.